This is Boston University's Neurophotonics podcast. My name is Sung An, a second-year PhD candidate in biomedical engineering, and I am the producer and editor of the podcast. This episode, our lovely hosts are joined by Dr. David Boas as he talks about the growing pains, the thriving present, and the optimistic future of Boston University's Neurophotonics Center. He also speaks on his journey to become the center's director and what makes this center so unique. So, thank you for tuning in, and please enjoy the episode.
Hello, everyone. Welcome to the first ever episode of the Neurophotonics podcast, created here at BU by trainees of the Neurophotonics Center. My name is Dora Balog. I'm a third-year PhD student in biomedical engineering, and I'm here today with my co-host.
Sudhan Duwadi, also third-year biomedical engineering PhD student, and we both are research trainees at Neurophotonics Center as well.
Okay, we are here today with Dr. David Boas, who's a professor of biomedical engineering and the director of the Neurophotonics Center here at BU. Welcome.
Yeah, thank you.
How are you feeling?
I'm great. Thank you. My computer just shut off. But I'm so happy that you guys are doing this podcast. So I really look forward to seeing all the future episodes.
Awesome. So thank you for coming, first of all. The first thing we're going to do is ask you to introduce yourself, tell a bit about your background and everything that led to this point until the foundation of the Neurophotonics Center at BU.
That's a long story. What confines do you give me? Constraints? I could go on for a long time.
I can start brief on where you did your undergrad. And
yeah,
then,
fine. I see. All right. All the way back there. And all right, I'll keep it brief. I was an undergrad at Rensselaer Polytechnic Institute in upstate New York. I was always interested in engineering. When I arrived there, though, I realized I didn't want just an engineering degree as an undergrad. So I switched to physics, something more fundamental. Really enjoyed my physics. Also really enjoyed skiing. I skied a lot. Ran the ski club there at RPI for 2 years. I think I skied 100 times between my sophomore and junior year.
Wow, that's a lot.
But I also did well in school, despite that, or because of that, maybe. Then, okay, you got an undergrad in physics. What are you going to do? So I went to graduate school. And when I was in graduate school at University of Pennsylvania in physics, most of the students were, it seemed at that time, when they got their PhDs, they were going off to Wall Street to be financial analysts. I didn't really want to do that. So my backup plan was to be a ski bum. So I really enjoyed my physics graduate school days and didn't stress that nobody was getting jobs because I figured I would become a ski bum if my physics PhD didn't lead anywhere. But I was very fortunate. I was working between two great labs, Britton Chance and Arjun Yode, and they were starting this field of biomedical optics. So there weren't many graduate students in the field at the time. The field was advancing very rapidly. And I found myself at the end of my PhD, having published a lot of papers, I was able actually to find a faculty position here in Boston. And so I came to Boston a long time ago, and I've been here since. So I first was at Tufts University. I was there for 2 years before Mass General Hospital recruited me to the Martino Center, where they had 6 years before, done the first fMRI experiments. And so they were interested in bringing me there to bring in optical methods, in particular functional near-infrared spectroscopy, to measure brain function to complement what they were doing with fMRI. They wanted to better understand, they wanted optics to help them better understand the bold fMRI signal. And so that was the beginning of just great activities for me and a lab that grew up in the Martino Center. So we became the optics division of the Martino Center, many postdocs, and that then became faculty. And we had a very vibrant group for many years. And it quickly went beyond just the FNIRs to measure human brain function to complement what fMRI was doing. But it also started then diving into microscopy methods. So we could look in animal models at a smaller spatial scale to help us better understand the fMRI signal. I mean, that kept leading to the development of new microscopy methods to help us answer open questions. We'd address those questions, which would lead to new questions, which would then need new optical methods, and we'd have to develop those optical methods. And we also started working with colleagues who were studying stroke physiology. And again, they had questions that we could easily make new optical methods for to help them address in animal models. And so it just started this cycle of learning how to interact with neuroscientists broadly, although at Mass General Hospital is mostly translationally relevant applications, clinically translationally relevant applications. And so we'd understand what their needs were and then develop the technologies for that and continue the cycle. And that was very successful and a lot of fun, just to be able to develop something and see it have impact very quickly and then lead to new questions that needed new technologies. And so we did that cycle several times over. I was at Mass General Hospital 18 years. But then there was an opportunity at BU to come back to a more traditional academic setting with more students. It was always a little sad we didn't have many students at Mass General Hospital. Always had one or two, but not many. And so it was really great to have the opportunity to come to BU, to be placed back in college environment with undergraduates and graduate students. But also BU is just very strong in neuroscience and photonics. And when I was arriving at BU, photonics was always down the street in the photonics building, and neuroscience was 2 blocks the other way down the street in the life science buildings. And, that 2 blocks was kind of a barrier. Despite there being like many faculty doing photonics work that could easily be adopted in the neurosciences, and many neurosciences needing novel optical technologies, right? That there was a barrier that some faculty were overcoming. But it was a great opportunity for me to come to BU and launch the Neurophotonics Center just to kind of more formalize and facilitate more interactions between all this faculty.
What did the early days look like of the Neurophotonics Center? So when you started, how many faculties were there? What were the programs you guys were pursuing?
Yeah, it was funny. Interestingly, I thought when we launched, I wasn't even at BU yet, so I wasn't fully aware of the full landscape. I thought we'd have a dozen faculty, but that quickly grew and grew and grew. As you can see on our website here, we have now over 50 faculty in the Neurophotonics Center, right? So it's just a tremendous array of people, and these people are all very active and engaged at multiple levels, within their Neurophotonics Center. So that was just a pleasant surprise for me. I mean, in hindsight, I should not have been surprised. But yeah, initially I thought it'd be like 12 faculty, but it's over 50 faculty.
That's awesome.
Yeah.
We always wondered, like, there's so many things you're doing. Mm-hmm. How did you end up doing all these things? So it was nice to know all the trajectory from like fMRI to developing new methods to answering new questions that led to the new methods and so on.
Right.
That was awesome.
Yeah, we were just super lucky. And I mean, mind you, this was over 20 years ago now, right? So there's just a lot of low-hanging fruit for developing new optical methods to address these outstanding questions in neuroscience. And I think that speaks just broadly to all of neurophotonics, right? Over the last 20 years has just had tremendous explosive growth because, of the ability to exploit all of these new optical technologies for the benefit of intravital imaging, mm-hmm. right? In the neurosciences.
So after you joined BU, you continued basically all of the research that you were also doing at the Martinos, right? So can you give us a brief introduction to some of the methods and techniques and research that you are still pursuing?
Mm-hmm. Interestingly, the year I arrived at BU, 2017, this was the 1 year in my independent research career that I essentially didn't have funding for functional near-infrared spectroscopy.
Oh, wow.
Yes, that was very sad. We had previously launched the Society for Functional Near-Infrared Spectroscopy. We trained over 1,000 researchers over the prior 17 years in how to use FNIRS, and we had no more money. But I did have good grant money for microscopy applications, and so I was pursuing that. And I was a little concerned that we may not get funding. But then the first grant I got at BU was to build a wearable FNIRS system, and we actually got a score of 10 on the grant from NIH, which is a perfect score. So that was a small grant, and it allowed us to prototype a wearable FNIRS system. I forget how many years that was. It was maybe 4 years. But before that grant ended, we then wrote a new, bigger grant within the Brain Initiative program to build actually kind of this system you have here. And we got that awarded. So coming to BU, was my FNIRS research ending, but no, it launched again. And now we're in this entirely new direction of doing high density, whole head FNIRS measurements of human brain activity, and just trying to, now that technology is working, right, we're trying to figure out how can we actually use this in everyday world settings to study brain activity in new ways, right? So that's a big focus for me and my lab. And of course, we also continue the microscopy stuff. And we have very good translational activities going there as well. Yes.
What are some of the other activities the faculty at the Neurophotonics Center is working on?
Yeah, many. We could spend a long time talking about that. So on the FNIRS front, we're very lucky. We have Miriam Uchel, who is in part of the Neurophotonics Center, technical director of FNIRS, and she supports 10 different faculty at BU and their trainees in adopting FNIRS for a wide range of applications. I have a nice slide here. These are most of the faculty. I probably missed one or two, but really nice examples with Lou Awad and his trainee Regina Slotsky, where they're doing gait rehab during post-stroke recovery with an exosuit, and they're kind of using FNIRS to help them better understand the walking efficiency and how that's improved with an exosuit. And our colleagues Terry Ellis and Alice Cronin-Gollum looking at Parkinson's disease. For instance, Swati Kiran is looking at aphasia and brain trauma. Just to mention a few examples. It's exciting just to see how FNIRS is being adopted for those many applications. Many of them are neurorehabilitation. And so I think that's an exciting field that BU can play a strong role in advancing. Also on the microscopy side, BU has had made a lot of impact in developing voltage sensing approaches. So using voltage sensors and the imaging methods, so you can, in animal models, measure just membrane voltage potential. So previously it was all with, electrophysiology. You get point measurements, or maybe you have arrays of measurements, but it's not an image, right? But with these voltage sensors, you can get images. But this really high speed imaging is required, the low photon counts, right? So severe technological challenges. And, many BU faculty have made significant advances on the technological side, and then working with other BU faculty to do cool new neuroscience applications with that technology. Those are exciting areas, right? Just to name a few. I'm sure I missed like a dozen more, right?
Yeah. One of the adjacent, I think, is SCOS. Do you want to touch on the SCOS as well? It's pretty close to the FNIRS research.
Yeah. The SCOS is speckled contrast optical spectroscopy, right? So that is a big new direction for us. That's kind of exciting. It actually goes back to when I was a graduate student. Half of my PhD dissertation was actually measuring blood flow using essentially a laser Doppler method, except we were doing it through thick tissues. So we were detecting tens of thousands of photons a second. It was a very weak signal. So We had to use very fancy instrumentation to measure essentially the Doppler shifts of light from moving red blood cells. We couldn't actually measure Doppler shifts. So we had to look at intensity fluctuations and do an intensity autocorrelation function of that. And like figuring that all out was like I said, half my PhD dissertation. So I was always interested in whether or not that could be applied to measuring cerebral blood flow. And I can remember actually for several years after I got my PhD and I was pursuing my own lab and I was developing FNIRs, people would ask me, well, can you measure blood flow? I said, yeah, I worked on it, but it's never going to work for the brain. We just don't have enough photons. But then, my colleagues back at the University of Pennsylvania showed that you could actually do it. And I was stunned. So we were developing that technology afterwards. And, my colleagues at Mass General have pursued many, pretty interesting clinical applications for measuring cerebral blood flow using light, right? Essentially laser Doppler. But it was not conducive to measuring brain function. The instrumentation was expensive. In parallel, though, in mice, we had been using something called laser speckle imaging. That was a cheap camera, didn't need photon counting, and it could give us a map of blood flow in the exposed mouse brain in 5 milliseconds. It was really cool. Subsequently, other people showed that you could actually use this speckle contrast method for human measurements of blood flow. And so we finally jumped into it maybe 5 years ago. And we realized for the first time that this actually works if you have fibers delivering the light from the human head to a camera. We first did it with a $20,000 camera. We then did it with a $500 camera. Then we did it with 16 $500 cameras so we can get an image of cerebral blood flow in a human. And so that's been really cool because basically, instead of measuring hemoglobin concentrations as we do with FNIRs, as this device does, we're using speckle contrast with these $500 that we detect with these $500 cameras. With FNIRs, it took us 20 years to go from measurements with single channels to measurements with high density arrays of channels. And with that speckle contrast, we were able to do that basically in 3 years. So that's been a lot of fun. We'll see where that goes. I think that has great potential for much further technological development.
That's awesome.
So I would like to pivot a little bit into the organizational side of the Neurophotonics Center. So Dan and I have both been trainees at the Neurophotonics Center. We have taken part in the training program. So Can you tell us a little bit more about how these boot camps are being organized? What is the purpose for them? We both had very nice experiences, but we would be very happy to hear from you as well.
Right. This comes back, I'll back up a little bit to the mission of the Neurophotonics Center, right? So the mission of the Neurophotonics Center is to build and support this interdisciplinary community that develops and broadly deploys photonic technologies, to advance our understanding of how the brain works, right, in health and disease. So, we want to build and support a community to do that, right? So we need the trainees and the faculty. And so it's very natural that we would have a neurophotonics research training program. And we were very fortunate. Actually, before I came to BU, Tom Bufano, who's director of the Photonics Center, he wrote a grant proposal to the NSF to get a research training grant for neurophotonics. There's always funny thing. He called it the Neurophotonics Research Training Program. NSF calls it the NSF Research Training Program, so NRT is actually NSF Research Training Program, but we kind of borrowed that to call it the Neurophotonics Research Training Program. Causes lots of confusion for some people. So that was awarded essentially when I arrived at BU, which was exactly when we were starting the Neurophotonics Center. So that gave us a nice focal point to grow a trainee community around, the faculty who are going to be part of the Neurophotonics Center. And that just worked incredibly well. So we've had, since 2017, we've had, in the first years, it was like on average 15 to 18 students joining every year as they join at the end of the first year. in the last 2 years, we've had almost 30 students joining, right? So really, I think, the students are finding this a great opportunity for many reasons, right? One is all the training they get. So we start with a boot camp, right, that you all went through. And that boot camp kind of introduces them to the basics of neurophotonics in intensive 3 days. It also, I think this is the best part of it, kind of builds that cohort. And that's important because the trainees are coming from 8 different departments, right? It would be very hard for them to meet otherwise, right? And it's important that they meet, right? Because just neurophotonics and neuroscience in general is really a very interdisciplinary field. It requires people from many different skills and backgrounds to have a successful research program. And so this boot camp, allows the first years to meet other trainees from the 8 different departments, right? And then sustained throughout the research program, training program, they continue to interact with these students. We have regular socials. We have many other training opportunities that they engage in. And I think that keeps drawing in more students, right? Because they really appreciate those aspects and opportunities to interact with a broad group of people.
Right. So we were talking about the bootcamp, and this year we also had a data analysis bootcamp, which happened a few weeks ago. And Sudan and I had the pleasure of interviewing Ryan Rout, who came from Seattle to do this bootcamp with us. We had an awesome conversation with him. So we're just wondering, for the audience, can you explain a little bit what, other than these bootcamps and socials, what other student committees does the Neurophotonics Center offer? Because there are a few student-led committees.
Right. Yeah. Fantastic. So first, the data science aspect, right? So originally, the boot camp was just at the end of the first year, an introduction to neurophotonics in general, right? When the NSF training grant ended after 5 years, we organized ourselves and went after an NIH training grant, a T32, from the NINDS. And we were just awarded that T32 grant this past summer. And fantastically, that program, when we submitted the grant proposal, really required us to have an emphasis on data science and actually gave us funding to enhance the exposure to data science elements for the trainees. And so we're doing that in a couple ways. So one way was we added a boot camp at the end of the second year. So the students come back in their cohort and they get exposed to many basic elements of data science. I mean, this year we did actually a lot of just coding basics and good practices with coding. And Emily Stevens has been running that. And from all perspectives, I think it was just a really huge success and the students really enjoyed that. We are also continuing a monthly neurotechnology tutorial series, which is now expanded to include data science. And so the students all participate in that and get exposed to, great neurotechnology and data science. And so the other activities, so I always wanted the students to kind of take ownership and lead activities that interest them. And so I've always been encouraging the students to do that. They manage the neurotechnology series, tutorial series, and the socials that follow that. A group of students came to me a few years ago and said they wanted to run a neuroethics program. I think that's in its 3rd or 4th year now. So that's fantastically successful program. They have a great event coming up very soon. And now we're seeing more and more students come forward with ideas of their own, like you two. You wanted to do a podcast series, right? So that's a great activity for the Neurophotonics Center to support. So, students aren't required to kind of take on leadership positions like this, but I think a fun aspect is that the Neurophotonics Center can encourage and support that, and that some students are really happy to step up and do these extra activities because they enjoy it. And I'm sure in particular they enjoy that all their peers, enjoy it and benefit from it as well. So, I'm really happy that a small percentage is so highly engaged in this way. And also, so this year, kind of, recognizing just the need for the students to have more autonomy and power and control over the management of that, we started this neurophotonics training organization, right? So we just want to empower the students to really have kind of full control over that. We'll kind of give the student group kind of a budget, and we'll try to stay hands off as much as possible. But money is never free. You always have to provide some reporting. But I think that would just help just expand the number of programs. Although I'm already very happy with the number of programs or committees that the students are leading, right, for enriching the neurophotonics training opportunities. Does that address?
Yeah.
Yeah.
That's perfect.
Yeah.
That's perfect. And to attest to that, we trainees are very grateful that we have all of these committees and neuroethics seminars, boot camps, and guests from Allen Institute, like Dr. Ryan Routh coming here all the way from Seattle to give us knowledge on how to do computational analysis of neuroscience. We're very lucky in that regard.
Yeah. No, I'm glad you mentioned that because I forgot to elaborate on that, which is, so the NIH T32 also gives us funds to each semester have a quantitative skills workshop, right? So it gives us funding to encourage an external scientist to come spend 3 days with us, right? Give a general seminar, but more importantly, spend, quality time with the trainees with more in-depth, like data science workshops, right? Around some specific technique.
Yeah.
Right? And so that was our first one, right? I'm glad to hear the feedback that it went well. And you guys will work with Emily Stevens to organize that each semester, right?
Yeah, absolutely. It was great networking opportunities where we had opportunities of one-on-one lunch sessions and everything.
Right.
So that was great.
Yeah, super.
You touched on the T32 grant that you that the center recently got. So what are some of the benefits, like supporting the students or travel awards and those kind of stuff that the trainees would get from that?
Right. The T32, in addition to supporting those data science elements, provides funding for some stipends. And so at the end of the second year, students can apply for fellowship from the T32. And so we're able to make a small number of fellowship awards from the T32. And so students appreciate that, the PIs appreciate that because they don't have to pay this student from their own grants for 2 years. Right. But that actually, from my perspective, is a small benefit of the T32, right? The T32 is this nucleus that attracts the community. The center, actually not through the T32, but the Neurophotonics Center with support from the university, we're able then to, provide all trainees, travel award support, right? And give them these extra benefits for being part of the community.
Awesome.
Right.
I have also heard from some of the peers that they were able to go to the summer schools from Neurophotonics Center support. Would you care to elaborate a little bit on that and maybe future opportunities?
Yeah. I mean, we're always looking for ways to further enrichen the training opportunities for the trainees. And so one is this relationship we have with the Neurophotonics Summer School in Quebec at Laval University. And it's highly competitive to get into. But, we're able to send, 1 or 2 students every year to that. And so NRT students are able to apply for that, right? So that's a nice opportunity. And we're always looking for more opportunities and we'll take, input from the students as well, what we should be reaching out for.
Is Boston University having its summer school still?
Have we ever had a summer school? Anna DeVore really wants to lead a summer school, right? We have to raise funding for that. So we're in the process of raising funding for that. We will no doubt succeed, right? We want to do it. But you need good resources to do that. So
there now. We're looking forward to it.
Yeah. So the local trainees, will have to help run it, right?
Yeah.
We'll be happy to.
Right. And then, but in return, you get to interact and network with all of the outside trainees who will come in for the summer school, as well as the outside teachers, right? Scientists who will be teaching.
Yep.
Yeah.
Awesome. So looking into the future, what plans and visions you have for the future of the Neurophotonics Center?
Mm-hmm. The first 5 years was just explosive growth because there's all this opportunity to exploit by just different faculty getting to meet each other from, essentially 2 blocks away. So we had several lunches in the beginning and for the faculty and always new collaborations would form out of that. So that has now stabilized. The community, we know each other mostly, but there's always new faculty coming in. And so when the new faculty come in, I think it's a great attraction for anybody who is in a semi-related field to neurophotonics, because we get to plug them into this large community right away. Tremendous benefit for them to launch their new lab. So those continue to be great new opportunities. And just over the for several years, this is just a list of junior faculty that have benefited from basically launching their labs in connection with the Neurophotonics Center. This is actually an old list. There's many new faculty since then. And so they're bringing us in many new directions that are very exciting. For instance, Meg Younger, working with mosquitoes. That's an exciting new direction, since prior to that, many have here work on rodents or songbirds. And so it's kind of fun now to go into invertebrates. I saw a whole new dimension for the neuroscience research, and she's going to be exploiting that for the neurophotonics to help her advance her science. Other directions, it's also exciting. Actually, we have all of these intravital microscopes that other disciplines can exploit. For instance, we have Hadi Nia here. He is a cancer biologist studying lung cancer, right? So quite distant from neuroscience, but the tools are directly relatable. And so, that's just one example. We have many other non-neuroscientist faculty who are benefiting from the community as well. Finally, what I'm personally very excited about is our neuroscience of the everyday world, right? That I was alluding to here. We really want to use this FNIRS technology to take our studies of brain activation from a laboratory setting out into the everyday world, right? So we want to study brain activity while people are walking, while they're interacting with each other, right? While they're perceiving the world around them, right? So that is hard to do in fMRI. You're inside the tube of a magnet. It's hard to study the brain during natural interactions when you're in the tube of a magnet, right? But with this FNIRS technology, we can do that, right? So that is a direction that I'm personally engaged in. That's going to probably drive, take the next 10, 20 years of my career to really fully develop. But what's great fun is, there's all these other faculty at BU and a growing list of faculty who are going down this path with us. So we're going to have a lot of fun developing this new field that we call neuroscience of the everyday world.
That is awesome. Do you have any advice for PhD students who are starting their graduate journey and who are interested in the Neurophotonics Center?
If they're interested, they should join right away, right? Join right away, get on our mailing list so you can see all the activities that are happening and get involved your first year right away, right? So low barrier to just get on an email list and, read those emails. So that gets you started. And then we can even help you find lab rotations to find your interest, right? And then if you like it, then formally join and we'll, and there's a curriculum that goes with it and much, much more. That's one bit of advice, just joining. Are you asking for other bits of advice?
Whatever you want to share.
Yeah.
Do you have any research advice, how to navigate multidisciplinary teams?
I think at BU, just navigating multidisciplinary interactions is very easy. I'm very excited at BU. Just the faculty are really interested and eager and collaborating with other faculty in their teams, right? We all recognize how much we benefit from those collaborative interactions. And so, yes, students, if they want to try to form a project that would join two different labs, go for it, try it. Funding is always, a challenge, but I think oftentimes that the funding challenge can be solved, right?
Yeah, I can attest to that.
Right.
That was exactly the path I took between you and Dr. Kamal's and your producer son and Dr. Matthias Stengel, which is
exactly
EFIS in human subjects to be here. So that's awesome.
Right.
One of the other major advantages that I see of the center is how all of the researchers and students can use all of the devices in the Neurophotonics Research Center. So I think that would be a good highlight and interest to prospective students and prospective faculty members that want to join the Neurophotonics Center. So would you care to elaborate?
Elaborate on the instruments. So that's a really good question. So, we do have, I name them all? We do have on our website, which is just bu.edu, Neurophotonics, we have our core facility. So under core facility instruments, here is probably an outdated list. We have many more instruments, but this gets you started, right? We have our FNIRS core instruments. We have 2-photon microscopy, optical coherence tomography, and many other instruments. The vast majority of the people here at BU are using either 2-photon microscopy or FNIRS, but we have many more instruments, and people should just reach out and talk to us to see the instruments, right? Or talk with their peers who can, who are already using the instruments, and they can introduce them to the instruments as well.
To close out, a little bit of a theoretical question. Where do you see the neurophotonics going in the future? What do you think is going to happen in the next 10, 20 years, in your opinion?
I think one thing we've really clearly shown here at BU is just how enabling a neurophotonics center can be. And the reason for that is because the technologies are so advanced, it's hard for any one lab to really be on top of all that is needed to be on top of in order to utilize these instruments, right? And, these are usually not just turnkey instruments, which is what core facilities normally have, right? So we have a core facility, but we're not really a core facility. We're enabling faculty and their trainees to utilize just advanced instruments that are not turnkey. And I think that's the next 10, 20 years, that's going to continue to be really important just because the technology is going to continue to advance. And the advanced instrumentation is just going to be even harder to utilize, right? So certainly what's an advanced instrument today in 5 years is going to become turnkey, right? But we always want to be at that bleeding edge of the new technology because neuroscience is always looking, to be able to measure more neurons, faster, to look at more brain regions, right? And, there's so much technological evolution needed to actually realize that potential. So that is where, just more cells, more brain regions, faster, better spatial resolution, right? So this is the mantra. The last 20 years is going to be the mantra the next 20 years.
Awesome.
Thank you very much for spending almost an hour talking about your cool research center opportunities for the students and the faculties as well.
Great. Thank you, guys. I'm looking forward to watching your future episodes.
Yeah, thank you. Awesome.
Thank you for listening. If you like this episode and want more like it, please take a moment to share it with anyone you think would enjoy it too. Next time, we will be joined by Dr. Ryan Rout, a Shanahan Foundation Fellow at the Allen Institute and the University of Washington, Seattle, as he talks about his academic journey and the problem of arousal. This episode was made possible by the support of Boston University's Neurophotonics Center and listeners like you. Thank you.